Today we will demonstrate how to use Papyrus conjugation kits. This video will show how to use the protein-to-protein conjugation kit, which utilizes amines on both proteins for attachment. The first step is to dilute proteins that you want to conjugate in the amine-free conjugation buffer to a 0.5 millimolar concentration. If your proteins are already in another buffer, you need to replace the buffer with the conjugation buffer. This is called buffer exchange, and we can do this by using the provided 10K molecular weight cutoff filters. To see more about how to perform a buffer exchange, click the link above. Once your protein has been buffer exchanged 3 times, the solution should look clear. You can now add 100 microliters of conjugation buffer to make the final 0.5 millimolar solution. Mix by pipetting and move the protein solution to another tube. We will now rehydrate the standard protein containing 1.2 milligrams as written on the label. To create a 10 milligram per milliliter solution, we use 120 microliters of the provided conjugation buffer. Pipette to rehydrate and mix. Remember to thaw the standard protein after adding conjugation buffer as well as the linker and quencher vials for 30 minutes before use. After thawing, it is important to centrifuge all tubes to ensure that nothing is stuck to the sides. You should now have protein 1 and protein 2 at 0.5 millimolar. Linkers 1 and 2 thawed and spun down, rehydrated standard protein, and another set of linkers 1 and 2 for your standard protein. Add 12.5 microliters of one of your 0.5 millimolar protein solutions to linker 1 tube, and add 12.5 of the other protein into the linker 2 tube. Add 12.5 microliters of standard into linker 1 and 12.5 microliters of standard into linker 2. Pipette to mix. Incubate samples at 37 degrees Celsius for 1 hour. Next, add in 3 microliters of the quencher into each tube. Next, mix the 2 linker tubes together by moving all liquid from linker 1 into linker 2. Pipette to mix. Do the same for the standard vial as well. Transfer linker 1 into linker 2. Incubate samples at 37 degrees Celsius for 1 hour. The conjugation is complete. You can now use the same filter columns as before to buffer exchange into your favorite protein buffer and analyze your conjugates.